Hello, my name is Maaz Haque, and I'm going to be presenting to you my science fair presentation, The Effect of Oral Antiseptic on Bacterial Growth. First, I'd like to thank God for giving me the knowledge to complete this experiment. Next, I'd like to thank my parents for supporting me along the way, assisting me in the project, and purchasing materials. I would also like to thank my science teacher, Ms. Siddiqi, for teaching me the appropriate lab techniques that I used in this experiment. Lastly, I would like to thank you, the judges, for taking your time out of your busy schedules to judge my presentation. The purpose of this experiment is to test the effect of certain oral antiseptics on bacterial growth. The reason I chose this project is because I wanted to see how potent these oral antiseptics are at killing bacteria, and I am also interested in pursuing the field of dentistry. The benefit of this experiment is that people can gain a better understanding of the best oral antiseptic. If one was to compare the inhibition of bacterial growth using the two oral antiseptics, Listerine Total Care and Colgate PerioGuard, then Colgate PerioGuard will inhibit more bacterial growth than Listerine Total Care. The independent variable I utilized in the project was the oral antiseptic. The dependent variable was the inhibition of E. coli bacterial growth. The control variables included the same petri dish type, the same bacterial species, the same setting, the same quantity of oral antiseptic, the same number of filter paper circles, the same incubation time, the same volume of each oral antiseptic, and the same amount of E. coli. I'd like to share some of my background research with you. Maintaining good oral health can prevent tooth decay, gum disease, and bad breath, and it can significantly reduce the risk of significant health problems such as stroke, diabetes, and heart disease. The enzymes present in saliva destroy bacteria in many ways, such as the growth and the inhibition of growth and metabolism, the degradation of bacterial membranes, and the disruption of important enzyme systems in bacteria. Porphyromonas gingivalis is a common gram-negative bacteria found in the mouth, and it is heavily linked to periodontitis, a disease that tears away the tissue and the bone that supports teeth. E. coli is a gram-negative bacteria that is commonly utilized in microbiology lab experiments, which is why I decided to use E. coli in my microbiology lab experiment. Listerine Total Care is an antiseptic rinse that prevents cavities, it restores minerals to enamel, kills germs of bad breath, it cleans the entire mouth, and freshens breath. The active ingredient is 0.02% sodium fluoride. Colgate PerioGuard is an antiseptic rinse that is recommended for use between dental visits for the treatment of gingivitis, which is characterized by the redness and swelling of the gums. The active ingredient in Colgate PerioGuard is 0.12% chlorhexidine gluconate. Upon application within the mouth, chlorhexidine can kill nearly 100% of gram-negative and gram-positive bacteria within 30 seconds by disrupting their cell membranes. The materials I utilized in this project included 10 petri dishes, E. coli bacteria 10 microliter solutions, inoculating lubes, Listerine Total Care antiseptic, the Colgate PerioGuard antiseptic, disposable gloves, safety goggles, a refrigerator, tweezers, a phone, a filter paper, a hole puncher, a permanent marker, a pencil or pen, a notebook, an incubator, an NaOCl bleach, paper towels, and sterile droppers. So the first thing I did is I gathered all my resources. Then I put on disposable gloves and safety goggles for the purpose of safety. Then I sterilized the lab and lab equipment before I started the experiment. I used the permanent marker to label 3 of the petri dishes E. coli with Listerine, 3 of the petri dishes E. coli with PerioGuard, 3 of the petri dishes E. coli only, and the final petri dish as negative control. I used the sterile dropper and tweezers to insert 3 filter paper hole punch circles soaked in PerioGuard into the first E. coli with PerioGuard petri dish. I repeated the previous step with the other 2 E. coli with PerioGuard petri dishes, as well as soaking the hole punched filter paper circles in Listerine with the E. coli with Listerine petri dishes. I didn't need to put in hole punched filter paper circles for the E. coli only and negative control petri dishes. I placed the petri dishes in an incubator at 30 to 34 degrees Celsius for 72 hours. I used bleach to clean the lab and the lab equipment I used to perform the experiment. I observed the growth and inhibition of growth after the 72-hour period finished, and I recorded the data. I regularly monitored the temperature in the incubator every 24 hours during the 72-hour period. So this slide displays the E. coli only petri dishes. As you can see by the green lines in the petri dishes, E. coli grew in these places. As you can see, there are no filter paper hole punch circles because it did not need it. This slide displays the E. coli with Listerine petri dishes. Like the last slide, there are green streaks of E. coli, indicating growth. There are also hole-punched filter paper circles, which were soaked in Listerine. As you can see, there's still green streaks that go through the filter paper circles, which indicates that no bacterial growth was inhibited. It basically allowed it to keep growing. This slide displays the E. coli with PerioGuard petri dishes. Like the last slide, it has green streaks indicating bacterial growth and hole-punched paper circles. However, what's different is that you can see small rings indicating bacterial growth inhibition, as you can see there. This slide displays a table that was used to chart out the data. In the table, it shows that the only petri dishes that showed zones of inhibition were E. coli with PerioGuard petri dishes. This is a graphical representation of the data, with only bars for the E. coli with PerioGuard petri dishes. The negative control petri dish, E. coli only petri dishes, and E. coli with Listerine petri dishes displayed no zones of inhibition. The only petri dishes that displayed zones of inhibition were the E. coli with PerioGuard petri dishes. The first E. coli with PerioGuard petri dish displayed zones of inhibition that were each 0.6 centimeters long. The second E. coli with PerioGuard petri dish displayed zones of inhibition that were each 1.0 centimeters long. The third E. coli with PerioGuard petri dish displayed zones of inhibition that were each 1.1 centimeters long. One potential source of error in this experiment may have been the unequal distribution of E. coli throughout each petri dish. Efficiency of E. coli distribution differed between each petri dish, which could have affected the results. Another error that could have occurred was the fluctuation in incubation temperatures, which may have been due to the opening of the incubator door a few times during the period. This may have caused changes with survivability, which could have altered the results. Saturation of soaking for the hole-punched filter paper circles was different between each one, which could have also increased or decreased the zones of inhibition. Caution: multiple petri dishes as trials, and many filter paper circles were used to minimize these sources of error. Also, Ms. Siddiqi watched over my experiment, and she made sure that these potential sources of errors were mitigated. In conclusion, the hypothesis stating that if one was to compare the inhibition of E. coli bacterial growth using the two oral antiseptics, then Colgate PerioGuard would be the stronger oral antiseptic, was right. The E. coli with Listerine petri dishes displayed no zones of inhibition, even though they were prevalent in the E. coli with PerioGuard petri dishes. Therefore, E. coli PerioGuard is the superior petri dish in antibacterial defense. The main ingredient was chlorhexidine, which was supported by the background research because of its effectiveness in killing bacteria. Thank you for listening to my presentation. I would also like to thank IJAS for the opportunity to present given this in the regional science fair.